Howdy, my name is Castillo, and my lab partner is Rita, Cantu, Mahurin, and this is our lab report on Lab 1, Handwashing, for VTPB 426, or Microbiology Lab. So how important is handwashing, and what is the purpose of this experiment? Well, handwashing is done so often that you don't really think about it. But if you do think about it, you think of how many things you touch daily. You don't know where those things have been. You don't know the bacteria and different microbes that are on there. And when you wash your hands, you're essentially getting rid of those microbes so that you are clean and you avoid any sort of infection or cross-contamination with anything when it's either handling with food, handling your face, handling your mouth, anything along those lines that you don't know where bacteria could be coming from. And hand washing is essential to remove those microbes from infecting something or infecting you or affecting your body in any sort of way. So it's really important to know and show how important hand washing is. And with that, we're going to use our materials and methods for this experiment, which includes 4 blood agar plates, cotton swabs, deionized water, a sponge, hand soap, saline, and most importantly, our hands. So our hands are going to be used, both of them, our left and our right. Our right one's going to be washed with only deionized water for 1 minute, then an additional 2 minutes, then an additional 3 minutes, adding up to a total of 6 minutes of hand washing with intervals in between so that we can swab and test their bacteria by inoculating them onto those blood agar plates. And our left hand is going to be used to wash with a sponge as well as soap to see the effectiveness of the soap compared to just the water. And that's going to be the same as the right hand, where it's going to be swabbed prior to washing, then for after 1 minute of washing, then after 2 minutes of washing, and then after 3 minutes of washing, for a total of 6 minutes of total washing with 4 different swabs for each state. And for the plates, they're going to be split into 2 halves, with each representing 1 interval of the washing. So for 2 of those plates, it's going to be the right hand. So those 2 halves will be R1, R2, and on the second plate, it's going to be R3 and R4 for those intervals. R1 meaning the initial swab of the right hand with the saline solution prior to washing, and R2 being the 1 minute, R3 being the 2 minutes, and then R3 being the final 3 minutes. And then it's going to be the same for the left hand. The left hand is going to be the initial hand without cleaning at all, just the saline solution. And then after 1 minute of washing with the soap and the sponge, and then L3 will be the sponge and water and soap for 2 minutes, and then the L4 will be the last 3 minutes. So all of these will be to test how effective the hand washing is with the soap and the water, or just using the soap in general.
For the results of this experiment, they truly showed the importance of washing our hands with soap and water, as it is vastly more effective than only utilizing water. When we only used the water, the bacteria didn't seem to cease to grow at all or reduce in any type of way. So the following photos in the next 2 slides are the results of swabbing Diego's hands after hand washing, and also were swabbed by myself. The results in this photo specifically are the plates that show the growth that was a result of utilizing only the water to perform the hand washing on the right thumb. So on R1, the left plate on the left side, after the inoculation of the right thumb without any type of washing, we can see that there's minimal colony growth that's visible on this portion. And then after rubbing the right thumb and pointer finger under the deionized water for a minute, the R2 portion of the plate was inoculated with the right thumb and many small colonies were present. On the right plate, the right thumb and pointer finger were once again rubbed under deionized water for 2 additional minutes, and we noticed that a similar amount of colony growth was visible after the inoculation, but there seemed to be a slight decrease in the amount. And on R4, the right thumb and the pointer finger were once again rubbed under deionized water for 3 minutes, and the inoculation on R4 resulted in a significant decrease in the amount of colonies. This slide is representing the results that were collected after using water and a soapy sponge to perform the hand washing on the left thumb. L1 on the left side reflects the inoculation of the left thumb before any hand washing was performed, and the colony growth here is minimal and scattered, but it is still present. After scrubbing the left thumb with the soapy sponge now for 1 minute, L2 is inoculated with the left thumb, and we can see an influx of small colonies were present on this portion. The left thumb was once again scrubbed with the soapy sponge for an additional 2 minutes, and this was shown on the L3 portion, and we can see a drastic decrease in the colonies that were visible after inoculation. And we did note that there was hemolysis visible. The left thumb was once again scrubbed for an additional 3 minutes, and the results on the L4 portion of the plate do show that there is no growth that was visible to the naked eye. Overall, we can see that the bacterial growth change over time with the water washing in comparison to the soap washing truly showed that there was no growth on the left thumb that utilized the soap, and it demonstrated how effective it is to utilize soap. In addition to this, we did see that there were some unexpected results on the L3 portion, which was the hemolysis that we could see, which is the ring that's visible around the colony. And this can occur for several reasons, such as contamination or improper sample handling. And this causes an unintended breakdown of the red blood cells, and it can interfere with the results we were collecting. In addition to this, limitations in simulating a real-world hand washing, there are a lot of factors that are different in a laboratory compared to real-world hand washing, such as not being as intentional with the time that is utilized to wash our hands, as well as this type of soap we were using. And there are a lot of different factors in the real world, such as the cleanliness of the area of the hand washing as well. The improvements for future experiments would definitely be being more intentional when utilizing the plates, because we did have some struggles with utilizing the biofilms in sealing our plates, and this could definitely contribute to any type of contamination. And in addition to this, we just want to make sure that we are ensuring that there are not any types of contamination so that our results are as valid as possible. And the way this experiment does connect to the real world truly shows that even in the elementary basic foundations of this concept of washing our hands that's taught to young children, it is definitely visible with this experiment, as we can see that it's an extremely effective method of reducing the spread of microorganisms that could possibly and potentially be pathogenic.
So in conclusion, soap and a sponge is way more effective than just using water to wash your hands. So unexpected results included some growths increasing as the inoculating occurred, but those could be explained by just technique on washing, on the inoculating itself. There could be different factors, because in theory, after time with the soap and the sponge, the reduction should be more and more, which it does happen in L3 and L4, but in L2 it doesn't appear that much of a difference. So it could be a reasoning behind that. It could be a reasoning because there's not enough washing, but there's just different reasons for it. So soap is more effective than just water because it actually breaks up the bacteria and removes what's broken with water. You rinse those broken down bacteria and microbes with the water that the soap used to break those down. And I didn't know that. I thought that prior that soap was just used to kill them in general. But no, it just breaks it down and essentially wraps around the bacteria so that it can wash off easily. But if you were to use hand sanitizer, for example, that is meant to kill the bacteria itself, then that would show much less bacterial growth than it did in much shorter time probably than in compared to just washing your hands. And there's benefits to using both, but hand washing is still incredibly important because there's some bacteria that can't be killed by hand sanitizer that you still need to remove. So you just wash your hands instead, and instead of removing it, you can rinse it off compared to the hand sanitizer that will either not kill it or it will kill it, but it'll also kill some other things that don't want to be killed. So overall, just hand washing is important, and so is hand sanitizing because the hand sanitizing will kill the bacteria. And this just procedure shows that hand washing is incredibly important of being clean and reducing the risk of infection. Rinsing the water only, it showed no reduced amounts of bacteria. It just stayed either consistent or like minor changes because of what could be technique or different factors that we explained before. But overall, hand washing with hand soap is very important because the water doesn't remove the bacteria in the way that soap does, because soap actually breaks down those microbes and engulfs them, removing them from the area just overall after rinsing.
Thank you so much for listening to our results on our experiment about hand washing. We hope you enjoyed it.